So first, welcome everybody to our first seminar in the spring. This is our first spring seminar. I think yesterday it really felt like today. But we're very happy to have Geoffrey Pitt from Columbia University from the Department of Pharmacology. And Jeff received his MD and Ph.D. both from Johns Hopkins University, and then across the country to the West Coast, where he continued to do work on ion channels in Dick 10, Richardson Laboratory at Stanford. He also, I think at Stanford, did his residency in internal medicine. So Jeff is now 2 months out of the year he does internal medicine. Or cardiology, general cardiology. I've gotten to know Jeff's work as it was published because he did some beautiful, really beautiful work on L-type calcium channels, and especially the interaction of calmodulin with the calcium channel and its inactivation. And this work was published in a series of fantastic papers from Jeff, together with Zulke and Harold Reuter from Bern, and also Dick Chen is on the papers in Schulman, just on one of them. So that was my introduction to Jeff's work much before we met. And we met actually last June when I gave a seminar at Columbia, and that's when I decided that I would love to bring him here and listen to his seminar. Recently, he has been working on IKS also, not just on calcium channels. Your seminar. Thank you.
So thank you very much for the invitation, Yoram. It's really a pleasure to be here. It's actually quite intimidating, too, for 2 reasons. One is I realize that I'm being videotaped. So any mistake will live on the web for perpetuity. And the other is that I realize that the data is fine, but my interpretation of the data I have to be very careful about, because whatever I say, one of the modelers will go back and in 5 minutes tell me whether I'm really right or wrong. So for those 2 reasons, I'll try to be careful what I talk about. And then just one quick story about internal medicine. Essentially, I'm a cardiologist, and all I do clinically is cardiology. I just did my 10-year recertification in medicine. And just so all of you know, when you go to your physicians, I passed without having done any internal medicine for 10 years. So just remember that when you see your physician. Cardiology. Anyway, the title that I gave quite a while ago is up here, Modulation of Calcium Channels by Accessory Subunits. And as I was getting on the airplane today and trying to figure out exactly what I was going to say, I changed it a little bit, but I'm going to stick to that general theme. And just to start, I wanted to show you our river. This is Columbia University Medical Center right here. This is the George Washington Bridge. So you're looking at the corner of New Jersey right here, and then this is Manhattan. So I'm going to talk about calcium regulation, and in particular we're going to talk about calcium channels today. And towards the end, I think there'll be a little bit of time, hopefully, I'll talk about some of our more recent work on calcium and calmodulin regulation of KC and Q1. But just to bring a focus to this, calcium, as you well know, is the kind of final common pathway of excitable cells. And so one kind of calcium-centric way of thinking about it is all ion channels do is regulate membrane potential. And so there's stimuli such as voltage or ligand-mediated channels, some sort of ligand that will change the gate, and that will change ion fluxes and change membrane potential. And this would be essentially fetal cycle, except for the fact that it's working. Is that excitable cells have calcium channels and therefore change internal calcium concentrations, and that modulates the specific effects. And in particular, looking at the effects of calcium channels, they're quite wide. Here's a bunch right here. So there's effects in the cardiovascular system that are really well in here. Multiple effects of calcium channels and their regulation circulation in the brain, and then there's controlled insulin secretion, so calcium channels are particularly important for diabetes. And then more recently there's misspelled, but I know that there's a problem on the left forever. That there's effects on immunomodulation as well as development. Focusing specifically on L-type calcium channels, the CAD 1.2, you see that they're expressed in a variety of tissues, very widely expressed, and have a wide variety of functions. And we know about this because of the recent identification of Timothy syndrome, which is a single point mutation defect in one domain, and there's two different mutations that have now been described. And these poor patients have a variety of defects, including effects of development. They have abnormal facies, they have syndactyly, they have long QT syndrome, they have problems with insulin secretion, they have problems with their immune system. This is an extremely rare event. I think there are probably more people in the world that study this disease than there are that have it. But it's been quite interesting in terms of giving us information about what calcium channels do and about how calcium channels work. With this kind of setup of how important calcium and calcium channels are, it's not surprising then that for all channels that calcium can feed back and then modulate the very gates that are eventually regulating the calcium channels. And so you can have inhibition of these gates as color-coded here, or you can have activation of these gates, and both directions work. And I'm going to give you an example of both of these today. Yoram already mentioned some of the work that we have done on calcium-dependent inactivation of calcium channels, and I'm going to talk to you about some newer work that also kind of dates back from the end of my time in Dick Shem's lab, talking about calcium-dependent facilitation of L-type calcium channels. And then I want to try to make a more general statement about what calcium and calmodulin do for calcium channel, or for channel function in general, in terms of regulating channel biosynthesis. So this is, to start with the calcium channels, this is a schematic of the pore-forming subunit of the calcium channels. And you can see the pseudo-fourfold symmetry with 4 sets of 6 transmembrane domains linked by these intracellular linkers. We now have structure of beta subunits, at least in the core domain, and we've done some work on beta subunits, and I have been thinking about talking about beta subunits, but I'm not going to. But that's one of the other critical accessory subunits for calcium channels. And then what I want to convince you of today is that calmodulin we now think of as an obligate subunit. So this is essentially a subunit of the channel, and that has important implications that I want to talk to you about towards the end of the talk. But what I want to start talking about today is the role of CAM kinase 2. And as of last month, we now have a crystal structure of the catalytic domain of CAM kinase 2 that's shown here. And so what I'm going to start talking about is something called calcium-dependent facilitation, which is a feed-forward facilitation or increase of calcium influx through the L-type calcium channels. That in the heart is regulated by rate, so it's part of the force frequency relationship, and it's increased calcium influx in response to increased rate. And there's been obviously plenty of work on that done here, so I'm going to give you some of our more recent kind of biochemical and functional results. So this is the TREPI phenomenon, the force frequency relationship, which is well known. And what's plotted on here is the amount of force that's developed in a muscle strip against on the x-axis the time between stimulations. So as the time between stimulations or the frequency increases, as the time goes down, you can see that more force is developed. This is another way of plotting these data. Each of these lines represents here the amount of force that's developed. And so if you're stimulating once every 20 seconds, you develop about that much force. And as you increase the frequency, so it's 1 to 3.63 seconds, not only do you see a lot more lines, but you see more force generated that then returns back to a lower force generation at a lower frequency of stimulation. So there are several, at the cellular level, there are several things that contribute to this. One is that there's decreased calcium removal during a shortened diastole, and part of that is because terms of regulation of calcium uptake by phospholamban and SERCA. But another important part of this is that there's increased calcium influx. And so at faster heart rates, there's what I'm going to tell you about is there's phosphorylation by CAM kinase 2 of the L-type calcium channel, and that leads to increased calcium influx. And there may also be increased release from lorandinine receptors, also via CAM kinase 2 phosphorylation, which I'm not going to talk to you about today. So this is recordings of L-type calcium channels expressed in oocytes. What we did here are 40 successive depolarizations at 3.3 hertz. And we used a mutant form of the channel, which, based on some of the work we published last year, we now understand how it works. And it essentially removes inactivation. So it's a mutant that allows us, to reveal very prominent calcium-dependent facilitation.
I'm just interested.
Okay. And so what you see here is 40 successive depolarizations when we use barium as a charge carrier versus 40 successive depolarizations when calcium is the charge carrier, normalized to the first pulse, which is shown in this dotted line here. And you can see this prominent facilitation in this mutant that removes inactivation. And what I'm going to show you later is that when we remove the interaction site with CAM kinase 2, we completely block this calcium-dependent facilitation. And these are the same data plotted now with the 40 pulses showing the relative increase in current. So under these conditions, we see about a 40% increase in current by the end of the 40 successive pulses. This calcium-dependent facilitation, what I'm going to focus on, happens with the L-type calcium channels. There's very good data from other groups, for example, David Hughes looking at calcium-dependent facilitation of P2-type channels. So there are probably important roles in terms of short-term and long-term plasticity at the synapse, as well as in the heart for the force-frequency relationship. Certainly, at least in the heart, it's very clear that this depends upon the actions of this calcium homogenously-sensitive kinase, CAM kinase 2. So this is the recently published structure of CAM kinase 2, and it's known to be a dodecamer. So what is shown here is the catalytic subunit in this dark green with an association domain in the lighter green. And it's kind of in the heel and spoke formation. And there's 6 subunits on top, and there's 6 subunits kind of on the bottom with the 2 wheels, one on top of one another. And what's a little bit controversial or not well understood right now is how collapsed that wheel is. But that's not really important for today's talk. But showing you some older data with the reconstruction, this is again the 6 on top, and behind is 6 more on the bottom. And we can rotate this, and you can once again see this kind of wheel and spoke phenomena for a total of 12 subunits. And this arrangement is important for the ability of the kinase to function as essentially as a frequency decoder. Beautiful work from Howard Schulman. And the way that works is that if you stimulate with these calcium spikes as shown here, under kind of basal conditions, you activate the kinase, and then the kinase will decay. And if these spikes are far enough apart in time, then the kinase will completely decay, the activity will completely decay back to its basal levels before the next spike comes. So what you're doing is you're sensing the individual spikes here. But the kinase has an ability to essentially have a memory, so that if the spikes are close enough together, then the kinase does not have enough time to completely decay between spikes, and so you can have activity that will build up. And so in this way, the kinase is a perfect decoder of the frequency of calcium spikes, and so activity can then translate this increase in frequency to an increase in activity. And what this depends upon is this architecture that I've shown here, that depends upon calmodulin activation, calmodulin binding to 2 adjacent subunits, so that one subunit, when it's activated by calmodulin, because of calmodulin binding, as I'll show you in a moment, can phosphorylate not only its targets, but it can essentially phosphorylate an adjacent subunit, which is called autophosphorylation. So it's really not autophosphorylation, it's not phosphorylating itself, but it's phosphorylating one of the other monomers in this dodecamer. And so this is the way it happens right here in cartoon form. You have the active site shown in red, and then you have an auto-inhibitory helix that's shown in blue. And when calmodulin binds to it, it opens up that auto-inhibitory, or it removes that auto-inhibition, opens up the active site so you can get phosphorylation of targets. If you have two adjacent sites that are both occupied by calmodulin, so that is when there is either very high calcium or when the spikes are close enough so that there's not time for the calcium to decay fast enough, then you can get phosphorylation of a particular residue, and that essentially, in the adjacent subunit, and that essentially locks it into a calcium-independent activity, and in that way the activity can build up over time. And the new structure that came out just last month shows how this works in kind of beautiful molecular detail, in that there are two adjacent subunits with their auto-inhibitory domains right next to one another. And what you also see is that threonine 286, the autophosphorylation site, is at the hinge of this auto-inhibitory domain. So phosphorylation is going to essentially open up that hinge and not let it close and not allow the kinase to turn off. So in the heart, it's pretty well established by a number of groups. And here are single channel data from Mark Anderson's group that that calcium-dependent facilitation depends upon active CAMK2. And so these are single channel data in the addition of CAM kinase. And what you see is a shift in modal gating, so that you see longer openings, and you can see that the aggregate current is larger, and it depends upon active CAM kinase 2 because you can block it with ATP analogs that cannot be hydrolyzed. You can block it with specific inhibitors of the kinase activity. So this is probably the nicest single channel data, but there's very clear data that this calcium-dependent facilitation depends upon CAM kinase 2 and CAM kinase 2 activity. But how it works has not really been that clear. So we took the approach of trying to figure out how it works by first figuring out whether the calcium channel itself was a CAM kinase target. And so what we did here was express in HEC cells the alpha-1c, or the core-forming subunit of the calcium channel, along with the accessory subunits, and then did immunoprecipitation of the intact channel, and then did phosphorylation with CAM kinase 2. And what you can see is that the channel is a substrate, so it gets readily phosphorylated. This is incorporation of P32 right here, and that's blocked by a specific inhibitor of CAM kinase. Now, what was interesting is we noted that in these pull-downs, even though we washed the immunoprecipitated channel quite extensively, in these pull-downs, we noticed that there was a band migrating around 50 kilodaltons that was also phosphorylated. And this corresponded to an autophosphorylated CAM kinase 2. So it suggested that CAM kinase 2, after phosphorylating the channel, or in the process of phosphorylating the channel, was becoming associated with the channel. So before going on to this association, I just want to show you that we then made fusion proteins that corresponded to the intracellular domains. And what you see here is that both the N-terminus and the C-terminus are both targets of phosphorylations, at least in these GST fusion proteins. And both of these domains of the channel make sense as kinase targets. The C-terminus is also the target for pKa phosphorylation and SARC phosphorylation, both of which also lead to an increase in current. The N-terminus is believed to be involved in PKC regulation, although the data are a little bit less clear for that. So both of these make sense as targets for a kinase to increase the calcium current. But let me go back to the association. So first of all, from purified or from isolated myocytes, we're able to demonstrate that the kinase in the channel are associated. So here is again immunoprecipitation of the channel from myocytes. And this is a calmodulin overlay because we didn't have an antibody that we felt was good enough to detect the delta form of the CAM kinase. And what you can see is that with the calmodulin overlay, there's a single band that's migrating about 58 kilodaltons. And this is just a positive control for a purified alpha subunit showing that the calmodulin overlay works. So it looks like in myocytes, the channel and the kinase are associated. And what we also found that was interesting is that the association depends upon the state of activation. So here are fusion proteins corresponding to the intracellular loops, the same ones that I just showed you for the in vitro phosphorylation assay. And what you see is that when the kinase is not activated, we were unable to pull down any CAMP kinase 2. When we activated the kinase with calcium calmodulin, we saw association with some domains, in particular the N-terms, the 3,4 linkers. And then when we autophosphorylated or activated the kinase, then we saw association with a bunch of other domains. So it looks like there's a state-dependent activation with different domains of the channel, which may be important for its activity.
So these experiments were done with solubilized channel, is that right? So when you...
These experiments were done with GST fusion proteins expressing the intracellular domains.
And then you basically pelleted them after solubilization? Is that
So we make
correct?
the GST fusion proteins, bind them to the beads, and then we add purified CAMP kinase that's either not activated with just calmodulin, or allowed to undergo the autophosphorylation. And so allow them to interact with the purified fusion proteins on the beads, then wash them extensively, and then this is an immunoblot for CAMP kinase 2.
And the fusion protein was attached to the beads after solubilization in detergent, is that
Yes.
correct?
For most of
For
them.
most of those
yes.
experiments.
Yeah.
And how many proteins are associated with the ion channel balls attached to the beads? If you just do like a Silverstein or look at mass spec.
How many proteins are
Would be associated with the calcium channel.
from
just-
like a myocyte?
Right, from a myocyte, just looking, like with at SBS page plots with Silverstein or mass spec or.
So
any
those
sense of
experiments have
quantitative.
probably been done by like Kevin Campbell and Bill Catterall from a while. We didn't do those experiments. So in this case, we have purified protein from bacteria, so it's the only protein around. So in a myocyte, they're probably, certainly the beta subunits are going to be associated, the alpha-2 deltas subunits would be associated, calmodulin is associated, we've done those experiments. And this is an immunoprecipitation from myocytes suggesting that the CAMP kinase 2 itself is associated. So there are, it's a large macromolecular complex. ACAPs are associated from Bill Catterall's lab. So there are quite a few proteins, but I don't think the list is endless.
But I think it's a technical question. So in the immunoprecipitate, if you just stain with Comesi, what do you see? You just see a single band.
In the immunoprecipitate, you stain with Comesi, would you see a single band? No, you would probably,
So that's
This is the, right, so.
these are, Kevin Campbell did this a long time ago, so when he purified the kinase, that's the way, and Bill Catterall, that's the way they kind of identified the alpha-1, alpha-2 delta, and the beta subunits. So they wouldn't do immunoprecipitation, they did, with lectin purification, affinity columns. But yeah, so there are a bunch of channels, proteins that are associated. So I'm missing the thrust of your question.
I guess I was basically wondering what the frequency dependence was on the interaction of different potential partners with the protein.
Okay, so
And we've been fooled, like we're working in detergent solubilized fractions with certain proteins about things that tend to bind to these detergent aggregates of the channel and things that sometimes are specific versus non-specific.
right. Well, so those are both important points. I'll talk a little bit about the frequency dependence of the interaction indirectly. And certainly, working with GST fusion proteins, isolated domains of the channel, that's a huge problem. These are, in some ways, kind of ancient experiments. We've moved on, and I'll show you some work at the end if I have time for KCQ1, to say that we will never work with proteins that we need detergent for solubilization anymore. But that doesn't mean that these experiments are necessarily bad, because if you then go on and you do the proper controls, which I'll show you in a second, you can prove that these associations make sense. And so let me try to prove that to you. Sticking with the same method, which has its admitted flaws, we were able to isolate a domain within the C-terminus that was very close to what's called the IQ motif, which is one of the one of the important determinants for calmodulin interaction, which Yoram alluded to at the beginning of the talk, and then I'll talk to you again about in a moment. But very close to that calmodulin binding site, we were able to isolate a small peptide that was important for CAMP kinase 2 interaction. And so mutating 6 amino acids that were conserved between alpha-1C, which is the L-type channel, and alpha-1A, which is the PQ channel, which also undergoes the calcium-dependent facilitation, we were able to completely block that interaction between the CAMP kinase and the channel is shown here. So here's the almost completely blocked. So here's the mutant, here's the wild type, there's the GST control, and here's some quantification of that. And so here are some data that suggests that association that we were demonstrating in vitro with all the caveats that you're alluding to is probably important. When we take that same mutation and make it now in the intact channel and record the resulting currents, you can see the prominent facilitation that I showed you before, and now that's the same channel that is bearing that same, that has that 6 amino acid mutation that blocks interaction, and now you see that calcium-dependent facilitation is completely lost.
So you recapitulated the effect in an intact channel, right, without detergent solubilization.
Right. So these are just simple functional reporting, not simple, but these are functional reporting. And so there's no longer any detergent issues at all. So this is essentially correlating the biochemical effect with the functional. And not surprising, I don't think I showed this data. Not surprising, given the fact that the biochemical data suggested that there were multiple sites of interaction. Even though the CAMP kinase 2 effects upon facilitation were blocked, the channel was still able to immunoprecipitate CAMP kinase 2. So it's still associated, but that site within the C-terminus is critical for calcium-dependent facilitation. That interaction site is critical for calcium-dependent facilitation. Let me tell you why I think that's the case. So having mapped now one critical site on the channel for CAMP kinase interaction, we wanted to see where the channel interacted on CAMP kinase. And so as a first approach, we generated in hex cells, we expressed in hex cells all the different isoforms that we could get our hands on for CAMP kinase 2, and then did pull downs with the C-terminus. And you can see that whether you use alpha, beta, or 2 different deltas, the gamma for some reason didn't work, but essentially there was universal interaction of all the different CAMP kinase isoforms with the C-terminus. So it suggested to us that the interaction site on the CAMP kinase was in a very conserved part of the CAMP kinase, and so it was present in all the different isoforms. And so we then designed peptides that corresponded to the autoinhibitory domain. This is also based on work looking at CAMP kinase interaction with the NR2B subunits and NR2A subunits of the NMDA receptor. And what you see here is when you design peptides that correspond to the autoinhibitory domain, that they block the interaction. So here's no peptide, here's a control peptide, and you can see that 2 peptides that are essentially mimic the autoinhibitory domain, or a peptide that's based on, the NR2B subunit of the NMDA receptor, which also mimics that autoinhibitory domain, but they block this interaction. So what this suggests is, now here's the autoinhibitory domain right here, so it suggests that the channel is binding somewhere in this pocket, close to the active site of the kinase, because peptides that essentially mimic this autoinhibitory peptide block that interaction. So the data I showed you with the immunoprecipitation suggested that the kinase was associated with the channel, and that was important, and the mutation suggested that's important for function. So we considered that there were 3 essential possible models for how the channel and the kinase can interact and how that can lead to calcium-dependent facilitation. So one is that the kinase, now shown either on in red, off in red or on in green, associates only when it's on. And I apologize for anybody who's breaking colorblind. It essentially goes on and off as the kinase is turned on and off. The other possible model was to see whether it was similar to the NMDA receptor interaction, where the kinase associates with the receptor and then stays on and cannot be turned off. It cannot be unautophosphorylable. And the third model that we considered is that the kinase stays on, but the kinase can still be regulated. That is, the kinase remains associated with the channel, but can be turned on or off via activation of the kinase. So I'll tell you that it looks like the third model, and let me describe why. So first of all, it looks like that once you remove calcium, so I showed you before that calcium co-modulin was important for allowing the kinase to interact with the channel. But once you remove calcium, the kinase can stay bound to the channel. So here are the pulled-out experiments using this critical C-terminus as the interaction determinant. And you can see that if you remove calcium by adding a calcium chelator EGTA, that you can remove the kinase only if you add that during the binding part of the experiment. But once it's bound, if you add calcium chelator afterwards, you can no longer remove the kinase. So it suggests that the kinase binds to the channel and then stays bound to the channel, even when even at the basal spike levels, even when calcium goes down. The next part of this is that it looks like the bound CAMK2 can be turned off. So we do the binding experiment, and then we add a phosphatase to remove the autophosphorylation and turn the kinase off. And what you can see, look first at this panel, because this is an antibody against the phosphorylated isoform of the kinase, showing that it's essentially active in this state here. And what you see is that if you add the phosphatase after it's already bound, then under these conditions right here, you can turn it off. The calcium, as shown here, itself won't turn it off. So that means the kinase can be bound to the channel, and it can be turned on or turned off, but it does not dissociate from the C-terminus. And this shows you that the autonomous activity or the activated activity can decay over time with the phosphatase. And then once it's turned off, that we can then add calcium calmodulin after adding the PT1, the phosphatase to turn the channel off, turn the kinase off. We can add calcium calmodulin back to reactivate the channel. So essentially what this suggests is that CAM kinase 2 can bind to the channel and then can stay bound to the tether to the channel, but then be turned on or turned off. So it's sitting there as a dedicated calcium sensor and a frequency decoder.
So would you say that's consistent with two-point attachment?
That there's probably, well, we know
Or
that
more,
there's probably...
two or more point
right,
attachment?
Yes, yes. Because as I mentioned, when we make the mutation in the intact channel, we can still, that in this critical site that blocks facilitation, that blocks the functional readout of this effect, the kinase is still, we're still able to immunoprecipitate that kinase.
And that the second, third, or fourth mode of attachments are not accessible without first binding via calcium calmodulin stimulation.
Well, it depends on which domains. There's, yeah, well, you
So
always need
calcium
calcium
calmodulin is
calmodulin.
required to get it on.
Right.
Right? And then it's going to be bound to several new conformers. And then you could dissociate, right, get rid of the calcium calmodulin, presumably with EGTA.
Right.
But it's still going to remain attached.
It's still going to remain
And modulatable.
Right, right. So it's essentially a tethered frequency, but it can be turned on or turned off. It's still accessible to the phosphatase. So it can be turned on or turned off once it's tethered.
And the other site, which goes back and forth with the calcium calmodulin, would also be reversible, right?
We think.
Comes off, goes back on.
We haven't really begun to explore the other sites yet. That's obviously a...
And how would it dissociate? How do you imagine it dissociates if not for calcium?
We don't think it does dissociate. We
Ever?
think it stays
It
on.
must sometime.
It's always there.
We think it stays on because, you know, the immunoprecipitation, we didn't do anything special to activate the cells. I mean, so the association is an artifact of our biochemistry in order to take these two purified proteins and put them together. But we think that in a myocyte that they're already associated.
Can you measure the time course of dissociation once they're isolated?
Of dissociation. Well, probably, anything can dissociate is probably very slow, I would imagine.
I mean, it doesn't disappear in 10 minutes or an hour or...
Well, I mean, over the course of immunoprecipitation, which is done in the presence of calcium, so that, you know, we haven't done those experiments in the presence of EGTA, except for the isolated domain of the C-terminus. But the whole channel, when the immunoprecipitation is done in the presence of calcium, it remains bound. You know, the immunoprecipitation takes, you know, an hour, 2 hours.
So Jeff, you think that the 6 amino acids on the channel is one of the binding sites for the model?
For the campaign.
Yeah.
Yes, yes, it's it's the binding site, or at least one of the binding sites, because we haven't identified and mapped the other binding sites. It's one binding site. We started with the C-terminus because of correlations with pKa and the SART and the critical role of the C-terminus because of calmodulin. So it's one binding site that when you mutate that, you can block the functional effects. So I think that's probably orienting the kinase so that it can then be an effective kinase to phosphorylate specific targets.
Is it possible that it changes the conformation of the structure that it doesn't bind, so that it's not a direct binding site.
Say that again?
So your mutation
Uh-huh.
may change the structure of the peptide that prevents the binding. But it itself, per se, is not a binding site. Is there any experiment to exclude that as possibility?
I can't think of any off the top of my head. I mean, unless we had kind of a co-crystal structure or whatever showing the peptide actually bound and those amino acids actually contacting it, I don't know. I mean, we clearly show interaction with a small peptide that contains those 6 amino acids, and when those 6 amino acids are mutated, it no longer binds. And it's in a region that's also shares homology with the autoinhibitory domain, but I cannot formally rule that out. So this is our kind of crude model of what's going on, is that the kinase is associated with the channel. The critical phosphorylation sites are in the amino terminus and the carboxy terminus, and that the kinase somehow is interacting with the active site, but the kinase itself can be turned on or turned off, and it kind of stays tethered. So it makes sense for the kinase as a frequency decoder to be associated and remain attached to the channel in order to be able to respond to these calcium spikes. You wouldn't necessarily want it coming on or coming off, at least our data are consistent with that model, with all the caveats that have been used. But let me change gears now and move on to calcium-dependent inactivation very quickly, just to show you that that's an example where calcium can feedback and modulate gates in an inhibitory fashion. And to remind you, this is published work now that this depends upon calmodulin interacting with the C-terminus in a region that's very close to where I just showed you the cam kinase 2 interacts. And this is the effect that we're looking at. So if you record calcium currents and you use calcium as your charge carrier, you see relatively rapid activation of the calcium, and then you see decay of the calcium current while you're still holding the cell at depolarized potential. Now, if you do the same experiment on the same oocyte, you just switch from an extracellular solution containing calcium to an extracellular solution containing barium, then you see that the barium current decays much slower. And this alteration of inactivation or decay in current with calcium versus barium is a measure of this calcium-dependent inactivation phenomena, which is important for regulating the length of the action potential. And this problem has been worked on for a quarter century, as you already mentioned, in a series of studies in collaboration with Howard Reuter and Roger Zolke in Howard Reuter's lab. When I was in Dick's lab, we discovered, as well as David Yu's group, discovered that calmodulin was the calcium sensor for this process. And so this is calmodulin. I've alluded to it already. It's an interesting molecule in that it has 4 calcium binding sites, 2 in the emitter terminal lobe and 2 in the carboxy terminal lobe. These calcium binding sites are called EF hands because of the way it looks in the structure. And then there's this long alpha helix in between these 2 lobes. And as just to go over very quickly, we know that calmodulin is critical for this process because if we express a wild-type calmodulin, we see this robust calcium-dependent inactivation. But if we express a mutant calmodulin, in which we've now mutated 3 of the 4 EF hands, this is essentially calcium unresponsive calmodulin, then you can see that most of the calcium-dependent inactivation is lost. It suggests that calmodulin mutants can act as dominant negatives to block the binding of endogenous calmodulin and block the calcium sensitivity and therefore remove this accelerated inactivation in the presence of calcium. So going over some more recent work that I now did in Columbia, is we were able to demonstrate that calmodulin is an obligate subunit of the channel. It remains attached to the channel. And it remains attached to multiple determinants in the C-terminus. And there's one critical amino acid, this isoleucine at position 1654 in the cardiac isoform, that forms that has a kind of, the side chain is this large hydrophobic side chain that appears to interact with the determinants that eventually inform the 1,2 loop, which is thought to be kind of the hinge lid, kind of like a 3,4 link from the sodium channels. And so when calcium enters the cell, you see this prominent inactivation via this calcium chain change in conformation and interaction with the 1,2 loop. And so this is all published data.
So calmodulin interacts with the channel even in the presence of EGTA? That's
Yes.
what you're saying?
Yes.
So that's sort of like not so consistent with a lot of the earlier biochemistry, which probably is wrong, which said that most of calmodulin in the cell was cytosolic, at least in the myocyte. They like to try and claim that 90% of it was floating around free in the cytosol, and the rest of it was like next to the junctional sarcoplasmic reticulum.
Well, I mean, calmodulin is a
very
highly
abundant,
expressed.
right.
So you don't. I mean, if 10% were not, not in the cytosol, then there's still plenty to essentially coat all of the calcium channels. So we have very good data biochemically as well as functionally that suggests that calmodulin is a constituent subunit. So let me show you where I'm going with this in a second. Kind of summing up what I showed you about calcium and calmodulin, I showed you that CAM kinase 2 is a tethered frequency detector, and that calmodulin, I didn't really show you, but just went over this very quickly, controls calcium-dependent activation. And then skipping over some recently published work, I mentioned that calmodulin is a constitutive or obligate channel subunit. And so where I'm going with this is that I now want to try to convince this audience that under these circumstances, when calmodulin is an obligate subunit, it can regulate channel biosynthesis. And so, as a first pass, I'm going to show you that calmodulin regulates channel trafficking, or L-type calcium channel trafficking. And so, this is the principle. So this is a slide from Carol Deutsch, that there's work from her lab and others that demonstrate that ion channel subunits associate very early during channel biosynthesis. And so for those cases in which calmodulin is a channel subunit, then to us it makes sense that it's possible that there can be this new mode of calcium regulation. Since the channel subunits are associated early, that calcium and calmodulin control any of these processes other than or in addition to control of mature channels that are inserted into memory. And so as a first pass, I'm going to show you that calmodulin controls trafficking of calcium channels. And why we started looking at this is that there was work from Marlene Hosey's lab that suggested that there was a determinant within the C-terminus that was important for targeting calcium channels to the plasma membrane. And this work was done in HEC cells and monocytes. And this work was published right around the time when we first realized that calmodulin was interacting with the channel in the same domain. And I said to myself, well, it's clearly interacting with the calmodulin binding site, and so calmodulin must be controlling trafficking. I have no interest in trafficking. I'll never work on it. And it's happened to me several times in my relatively short career. As soon as I say that, I end up actually having to do it. I'm going to show you that we actually have now explored how calcium and calmodulin regulate trafficking and prove this hypothesis correct. And part of it came out of when we were making all the mutations in this previous study from last year, looking at how calmodulin interacts with the channel. We noted that mutations that disrupted calmodulin interaction biochemically led to decreased current amplitude. These are a series of IV curves of the wild-type channel and two different mutants that disrupted calmodulin interaction. And we noticed that there was decreased current amplitude for any mutation that appeared to disrupt calmodulin interaction with this critical determinant, the C-terminus. So we took channels, tagged them with a GFP at their N-terminus, and expressed them into HEC cells so that we could eventually do, and I'll show you data where we've done that, where we could do not necessarily simultaneous, but correlated imaging experiments and functional experiments. And so here are wild-type channels, and you can see nice membrane localization of the GFP-type channel. And here are these two mutations, and you can see that there's marked retention of the channel in the intracellular domains, much less targeted to the membrane. We then performed similar experiments with these same channels in HEC cells where we co-expressed either calmodulin
or CAM 1234, which is a mutant where all 4 ENTMs are inactivated. So this is a completely calcium-insensitive calmodulin. And you can see that disruption of the ability of calmodulin to bind calcium led to decreased current amplitude. We then looked at gating charge, which is a nice measure of the number of functional channels, or an indirect correlate of the number of functional channels at the cell surface. And you can see that the CAM mutant had decreased gating charge when it was normalized for the cell size. And the reason why this is a little bit looks a little bit tricky is that these cells only were able to kind of survive and be healthy when they form these kind of huge aggregates. So the cell size for the CAM mutants were really large. So as soon as you normalize the cell size, you saw this marked difference.
Yeah. I'm wondering why the alpha charge is much smaller than the alpha charge.
That's typical for calcium channels. I don't know enough about it to tell you, but I mean, you always see that. And everyone else does too. I'm sure there's a good because calmodulin has multiple effects in cells, there's so much calmodulin as we've already alluded to, we wanted to restrict the role of the mutant calmodulin to the calcium channel itself. And so we made this construct where we took our GFP-TAG channel, and then borrowing a trick from David Yu, is we attached the calmodulin to the C-terminus of the channel by flexible glycine linker. And now we can make either a mutant or a wild-type calmodulin. And we know that the mutant calmodulin, its function is restricted specifically to the channels, not affecting other calcium-sensitive processes in the cell. And so first of all, if you look at the imaging experiments, here are examples. The linked wild-type calmodulin has nice membrane localization. The mutant calmodulin does not. The current amplitudes as shown here in the exemplar traces or the IV curves are markedly reduced, and then once again the same thing for the gating charges. So it suggests that you need to have calcium interaction with calmodulin, and calmodulin interacting with this critical domain in the C-terminus is identified by Arlene Hosey. That's important for a membrane targeting. And so this, as I said, kind of makes sense to me to kind of think of it as a slightly as a new paradigm, is that for those channels for which calmodulin is an obligate subunit, that calmodulin can therefore act as a calcium sensor to control any of these early processes in channel biosynthesis. And in particular for the calcium channels, I showed you stuff about trafficking, and then there's, as we've already alluded to, extensive data about calcium regulation.
Calmodulin is so important for cell function. Have you shown that when you using the calmodulin, other proteins like not directly associated with calmodulin also have trafficking problems, or they are trafficking abnormal?
Well, I think that our best data in terms of nailing down that calmodulin is important in this process is the experiments where we restrict the mutant calmodulin to the channel itself. Because if you overexpress a mutant calmodulin in a cell, then because calmodulin is ubiquitous and has is pleiotropic, then the mutant calmodulin can have multiple effects that you might find difficult to interpret. But in the linked constructs, then I think that we can safely assume that most likely we restricted the effects of the mutant calmodulin to the channel itself. And so I can't tell you whether in those cases we've affected other processes, but if I have a few extra minutes, I'll show you that another example in where we think that calmodulin is involved in assembly of JCQ1 channels, once again as an obligate subunit. So I think that this is probably a relatively broad paradigm for focusing specifically on channels, for those channels in which calmodulin is an obligate subunit, that multiple aspects of biosynthesis can be regulated. And so, as I mentioned, this correlates well with this trafficking determinant that was previously identified by Marlene Hosey, and more recent work that we're just trying to finish up now is we found another binding site in the amino-terminus for calmodulin that has also been found by Amy Lee, who is now at Emory and was at Bill Catterall's lab. And she's shown that there's competition between calmodulin and the calmodulin-like protein called calcium binding protein 1. And we've now shown that K-chip is associated with the N-terminus. And so K-chip is a calmodulin-like protein that is very important for trafficking potassium channels And so, although I don't have the data to prove it yet, it's very possible that K-chip could be regulating channel trafficking of calcium channels as well. But that's kind of speculative at the moment.
Does the new binding site have either an IQ motif or 1914 hydrophobic segment?
No, it doesn't actually. So
it wasn't predicted from the
it
primary
wasn't
structure
predicted.
at all.
It wasn't predicted, and so actually, Nathan Dascal was the first group. His group was the first group to suggest that Calmodulin bound the N-terminus. Because there wasn't a site, I actually didn't believe it until we did the, and then Amy Lee did it. So we eventually did it ourselves. But there's a very clear line. But there's a rash now of Calmodulin interaction. With lots of SK channel, for example, that does not fall in any sort of consensus binding site. So I feel comfortable because of the biochemistry data, which I don't.
There's only a pseudo-consensus site. It's just the easiest
Right.
one to find. There's
Right.
obviously many.
Right. And so to try to expand upon this paradigm, here's a quick list of channels that are regulated by calcium. And then here's a quick list of channels that are regulated by calmodulin. So it's essentially every channel that has been suggested to be regulated by calcium except for BK channels. Maybe calmodulin regulates them as well, but I haven't found that in the literature. So it's possible, and we've actually shown that calmodulin interacts with sodium channels, working as well on the NMDA receptor. It's possible that this is actually a pretty a broad paradigm.
Is there anything known about calmodulin involved in regulating gap junctions?
There is. So the people at Einstein, one of the gap junction centers in the world, they have some data that suggests David Spray. Yeah. So his group has some data that suggests that calmodulin interacts with gap junctions, and they have asked me for my constructs, and I gave them last year, but I haven't heard anything about it. Huh? No, it has not been published. Yeah, it hasn't been published, but there was a graduate student who says it is very nice data. I haven't actually seen it.
But the main regulation of gap junctions is extracellular. There's no calmodulin there.
Gap junction?
Yeah, extracellular gap.
And gap junctions are cell-to-cell communications. It's all
Extracellular. Intracellular?
I think almost all of the pH
Talk about two different caps. G protein activating protein versus gap junction, which connects two cells.
Talking about gap junctions.
Okay, so let me finish up on some of our recent work on casein Q1. And so casein Q1 is the core forming subunit for the IKS current, the repolarizing current. It's been suspected to be or postulated to be a calcium-sensitive current for many years, and how calcium regulates the current is not even known. There have been several recent reports suggesting that in the casein Q family members, of which there are 5, most of them are expressed in different neuronal tissues, that there are 2 IQ motifs, these very poor calmodulin consensus motifs within the C-terminus. And there's some data from casein Q2 and casein Q3, which together form heteromers that underlie something called the M current in neurons, and mutations in that current lead to epilepsy. There is suggestion that calcium via calmodulin regulates this M current in the brain. Casein Q1, as you well know, is the most commonly mutated locus in long QT syndrome. And there are these, as I mentioned, 2 IQ motifs that potentially are calmodulin binding sites in the C-terminus. But whether calmodulin binds to casein Q1 and what calcium and calmodulin do to casein Q1 has been quite controversial. So using some of the biochemical approaches that I alluded to, but I didn't have a chance to show you, we tried to explore whether calmodulin interacted with casein Q1, and it did what it did. And so in order to do that, what we did is we expressed in bacteria the entire C-terminus as a 6-histidine tag fusion protein. And now we have no detergent whatsoever. This is just sodium chloride, a little bit of tris, calcium, or EGTA when we want it. And what you can see is that in the bacterial extract, we can express the C-terminus quite well, but without detergent, none of it's soluble. And so we've seen this now for several cases in where we've looked at calmodulin binding motifs and channels, sodium channels, calcium channels. And so we use the same trick that we used in those cases, is now we co-express in bacteria
the C-terminus as well as calmodulin. And now you can see that all of the C-terminus is soluble. And then over a metal affinity chromatography to purify the 6-his tag protein, we can purify the C-terminus. And you can see that calmodulin purifies in what looks like a stoichiometric fashion. This interaction doesn't appear to require calcium, although I'm going to show you that it looks like calcium calmodulin has a higher affinity for the channel, because when we take the calmodulin mutant that can't bind calcium, then you can still see that the calmodulin co-purifies. So we were able to make this nice purified complex, no detergents, and to try to get an assessment of the stoichiometry of calmodulin for the C-terminus, because there are two IQ motifs. We took the purified complex and ran it over a gel filtration column, and what we saw is a single sharp peak that migrated at a molecular weight that's much higher than a predicted single one C-terminus and one calmodulin. The resolution is not that good here, and this is just showing you that in that peak you both have the C-terminus and calmodulin. The resolution is not that good here, so what we did is cross-linking with DSG, which is a homobifunctional lysine-targeted cross-linker. And here's the starting material. You see the C-terminus here, calmodulin's run off of the 7% gel. And after 5 minutes of cross-linking, all of the material is now migrating at a little bit less than 250 kilodaltons, which is now consistent with 4 C-termini and 4 calmodulin molecules. So essentially a 1-to-1 stoichiometry, a 4-to-4 stoichiometry. Now this is intriguing because it's been known that the assembly domain for casein Q1 channels is within the C-terminus. So we thought that we've now isolated a soluble, intact assembly domain that includes calmodulin. We know that calmodulin is a part of this because this is an immunoblot showing that calmodulin is now migrating in this higher molecular weight complex. But even still, we weren't certain of the stoichiometry. So what we wanted to do was determine whether one calmodulin was sufficient to generate this complex. So using this kind of link strategy, we made a C-terminus with a flexible glycine linker linked to calmodulin or CAN4-, and we're able to generate without co-expressed material purified fusion protein. So it suggests that one calmodulin with one C-terminus is sufficient to generate this soluble material. This material can be cross-linked to form a tetramer, as shown here. It doesn't prove that it's only one, but it suggests with these other experiments that the stoichiometry is probably 1-to-1. These are competition experiments that I'm going to skip over right now, but just look at the calmodulin is here and not here, and take my word for it, it suggests that calcium calmodulin has a higher affinity for this protein than the mutant calmodulin. So, as I mentioned, the assembly domain had been previously implicated in the C-terminus and had been identified to a region between amino acids 589 to 620. The numbers are not that important, but Dan Roden's group has recently shown that this region was probably not involved in assembly, but possibly involved in trafficking. So, having this nice system, we tried to address this by making a construct which now deleted amino acid 555. So, this putative assembly domain is now removed from the system, and you can see that we can generate the purified complex. It can be cross-linked, and under these conditions, we see what looks like a monomer. In other words, one calmodulin, one C-terminus, a dimer, or two calmodulins, two C-terminuses, and then a tetramer, 4 calmodulins and 4 C-terminus. And we run this over a gel filtration column. We see a little bit of aggregated material, but now we see 2 peaks, one that corresponds to a molecular weight consistent with the tetramer, and one that corresponds with the molecular weight of the dimer. So it suggests that the domain that was previously identified as important for assembly, that Dan Roden suggests is important for trafficking, is probably, as Dan Roden suggests, important for trafficking, because when we remove it, we can still see assembly in this domain. So it's still in the C-terminus, but it's just in a slightly different region.
I've lost track here. This is a KCNQ C-terminus you're looking at?
Yes.
Not a calcium channel?
Yes. Everything, we've switched to calcium cars. So the nice thing about casein Q1 is because it's rife with long QT mutations, so you have a bunch of mutations that you know are going to be important to trying to figure out what's going on. And so we use that information to look at mutations that were in either IQ motif 1 or IQ motif 2 to try to continue to address how many calmodulins were bound, where the binding site was, and what we're able to demonstrate was
what we're able to demonstrate is that a long QT mutation in IQ motif 1 prevented the ability of us to generate soluble material. A deletion construct that deleted IQ2 prevented us from even getting material that was expressed. And as a control, we looked at a long QT mutation that's just outside of IQ motif 2, distal IQ motif 2. And that one was still able to form a complex where we're able to get soluble material. So it suggested to us that long QT mutations within the IQ motifs disrupted the interaction with calmodulin. And so what is the functional effect of that? So here are wild type currents. Here are the currents with a mutation that's just outside of the IQ motifs. They're reduced, but still present. And here's an example of a mutation in IQ motif 1 that there's no current at all. So it suggests that there's a correlation between a mutation that disrupts calmodulin interaction and loss of function. So in order to look at this in assembled channel, we beating a horse to death. We've kind of used another catamer strategy where we now have either a wild type or a mutant construct, and just as a control, you can see that the wild type concatenamers lead to normal currents. And now we can make mutations where we mutate a long QT mutation in either one position or the other position. And these are mutations that we show biochemically can disrupt calmodulin interaction. And in any circumstance where it disrupts calmodulin interaction, the concatenamers are non-functional. So it suggests that based on the data I showed you before, that calmodulin interaction with C terminus is important for assembly, that in a dominant negative way, that those mutations lead to loss of current because of inability to assemble. And it doesn't matter whether the beta subunit casein component is expressed. So because of time, let me skip over to this very late, I apologize, to this last data. So if calmodulin is a sensor or is a subunit, is it also a sensor? And so here are data where we express the channel in oocytes, and then we inject calcium, and we inject calcium, and you can see that there is a time-dependent increase in the peak current in the casein Q1 channels, and in control oocytes, where no casein Q1 is expressed, you don't see any increase. This is important control because, as you know, in oocytes there is an endogenous calcium-activated chloride current, and we were able to block this effectively with myflumic acid. So it suggests that calmodulin associated with the channel is a calcium sensor, and that casein Q1 is a calcium-sensitive current and increases as calcium is raised. And so here are just traces from a oocyte expressing casein Q1 and a control oocyte. Going in the opposite direction, here are data where we expressed in an oocyte casein Q1, and then we record an IV currents, we record a set of currents here, just do the whole IV curve, and then in the same oocyte, we inject back the chelate calcium to below physiological levels. And you can see two things. One is you can see a reduction in current, and that occurs over all the test potentials within the physiologic range. And you also see the presence of this kind of marked macroscopic inactivation. So casein Q1 currents, as you know, are thought to have a hidden inactivation that's shown by the relief of this inactivation in the tail current. And by lowering calcium to subphysiologic levels, we think that we reveal some of this so-called hidden inactivation. Now, is calmodulin responsible for this? And so lowering calcium to the sub-physiological levels, is that what's happening here? The final example using catamers, I'm going to show you that I think that's the case. So here are just casein Q1 currents reported by themselves, and you can see this little hook in the tail current, suggesting this kind of hidden inactivation revealed by this step to minus 60 at the end. And then when we make a catamer, where we've now linked calmodulin to the N terminus of the channel, we can see that you still have the hook, and you can still see what looks like normal casein Q1 currents. But now when we make the catamer with a mutant calmodulin, calmodulin is unable to bind calcium. You can see two things. You can see the marked macroscopic inactivation and the beginning of the loss of the hook of the tail. So since it's late, let me just finish up by saying that we believe that calmodulin is a constitutive subunit
of the casein Q1 channel, and that calmodulin participates in assembly via its interaction with the C-terminus within a domain that has been previously shown to be critical for assembly, but in a slightly different region. And that long QT mutations that disrupt calmodulin interaction are dominant negative, probably because they affect assembly. And then these most recent data, which I just kind of figured out last week, so they're still kind of suspect in my mind, but they look pretty good to me so far, is that calmodulin and basal calcium appear to relieve this hidden inactivation. And so if we lower calcium by injecting a calcium chelator, or if we attach this calcium insensitive calmodulin, that we can bring out this inactivation much more. And then elevated calcium increases the casein Q1 current, possibly by relieving this hidden inactivation. And so now, having seen these most recent data, there are obvious experiments we have to go back and do to address inactivation specifically to see whether that's really what's going on. And that's obviously what we're doing next. So now I've shown you two examples in which I think that calmodulin as an obligate channel subunit can therefore be regulating different aspects of channel biosynthesis. I showed you examples of calcium regulating mature channels for calcium inactivation. But then with the casein Q1, it looks like it's important for oligomerization. And then for the calcium channel, it looks like it's important for membrane targeting and trafficking. So it's possibly a relatively widespread paradigm for those channels in which calmodulin is a subunit to add this new level of calcium regulation to channel function. And so these are the people in the lab that have done the work. Let me just mention a few people quickly. Most of the work on casein Q1 was done by Smita Ghosh, who was a postdoc in the lab. Some of the CAMK2 work was originally started when I was still in Dick's lab. It took a long time to finish. Howard Schulman and Andy Hudman, who as a Yale contributed to that aspect of the work. So thanks for your attention. Yeah.
I've got a question going back to the inhibitory peptides that mimic the L-type C-terminus. Is that correct?
The inhibitory peptides mimic the auto-inhibitory domain of CAM kinase 2, or the binding site on the NR2B NMDA subunit.
And so those block binding of CAM kinase to the L-type channel?
Yes.
But those also would interfere with the effect of CAM kinase on its other substrates in impact myocytes?
So one of those peptides is a peptide that should block CAM kinase activity. And so we didn't do any experiments where we looked to see whether that peptide blocked activity. Those experiments I showed you were just looking at binding interaction. So those experiments were trying to map the interaction site. But the AC3I peptide should block activity. And other people, so for example, Mark Anderson has shown that in fact that peptide, a single channel recording does block calcium facilitation.
So binding, like in the case of binding of the beta subunit rules the activation. Does it play any role for the
Well, so as I said, these data are really fresh, and so there's a lot of obvious experiments to do. So I'll tell you one thing, and I probably shouldn't say it, but I'll say it anyway.
We're not going to do it. Don't worry.
Yeah, that's okay. It's just more just kind of showing my ignorance. So we've had this data for a while, couldn't figure out what was going on. I can tell you this: when we attach calmodulin to the N-terminus, then the channel is no longer responsive to to casein E1. So if we express casein E1 by forcing the calmodulin, the mutant calmodulin, to be associated with the N-terminus, we can't get the normal increase in current.
There's no increase in the current.
There's no increase in the current.
The disk is still there.
Well, it's a little bit hard in oocytes because you always have a little endogenous, because there's an endogenous oocyte thing, but there's of an oocyte KVO QT1 white channel. But so, for example, under the right conditions, you can get a 10-fold increase in the current when you co-express casein E1, and you get a 0-fold increase in current when you put the calmodulin on there. So I think that there's something going on there, and the data is too fresh for me to figure out exactly what is going on.
What about any changes of kinetics? So the KCNU1 doesn't have any effect on channel, on kinetics or properties, not only.
It doesn't appear to be, but now kind of with this new hypothesis just generated in the past week, we have to go back and look at those data. What we know is that if you take a wild type channel and you express the KCNU1, you get beautiful IKS-like or KCNP1, KCNQ1-like current. And with the linked calmodulin, the mutant calmodulin, that doesn't happen. So we have to go back and even for without the KCNU1, we have to go back and now do protocols specifically designed to pull out an activation to test this hypothesis that I'm trying to build, which could be wrong.
Any other questions? Thank you very much.